I just wanted to start off by saying thank you so much to the organizers for selecting me today. It's a real privilege, and hopefully my contribution can add to already the stellar lineup that we've had this year. So I'm not going to spend too long going through the nitty gritty. I just want to get everyone orientated because I'm quite excited about a lot of things. So this talk today is going to be focused on some of the stuff we've been up to over the last 12 to 18 months. So a lot of the talk is going to focus on voltage-gated potassium channels. So these are 6 transmembrane spanning domain ion channels, which are comprised of the voltage sensing domain, so the S1 to the S4, and a pore domain, so S5 to S6. And these open and close, as I'm sure we're all aware, in response to changes in voltage across the cell membrane. So these are quite ubiquitously expressed throughout the human body, and they regulate lots of dynamic processes such as neuronal action potentials, cardiac action potentials, muscle contraction. They regulate vascular tone, gastrointestinal processes, and even T cell migration. So very, very important ion channels. So the ones that we predominantly study in the lab are the KV1s or KCNAs and the KV7s or KCNQs. And you can see here's a picture cartoon of a neuronal action potential. And when these channels get mutations in them, they can, or normally loss of function, but we are finding more and more gain of function as a community. It leads to hyperexcitability disorders, which can cause conditions such as epilepsy, ataxia, developmental disorders, and so forth. So there's a big drive to find openers of these channels. And we decided to take over the last 6 years a bit of a different approach in order to do so. And we've done that by screening or elucidating, as Colin highly pointed in my introduction, the elucidating the molecular mechanisms of medicinal plants in order to find novel small molecule activators. So here's just some of the lovely locations that we have visited over the last 6 years. And I'm sure you can appreciate the views. We certainly do. But a lot of people love this fact and say, oh, it must be such a great thing to go out and do. And for the best part, it is. However, we have had some trials along the way. There was a trip to Santa Rosa Island a couple of years ago where the arduous 10-mile hike up very steep inclines and declines, and we decided to do it with 50-pound backpacks. Once we got to the beach at the end, we were then promptly nearly washed out by the high tide, so we had to retreat into the entrance of the beach. It actually got up to about this rock formation here, so we were lucky. You might notice we're down to one tent. We lost the other one. So yeah, we've had our trials. Jeff likes to show this picture of me. He says it's me. This is taken about 20 minutes after we arrived after the 10-mile hike, and he says it's me looking out pensively into the ocean and admiring it. Actually, what I'm really thinking is whether I should just carry on walking into the ocean so I don't have to make the trip back in 2 days' time. And the reason for that, and I'm sorry if you're eating your tea or your dinner right now, or breakfast, but is my feet were absolutely destroyed. I decided not to show the underneath. This was taken a couple of days later. The underneath and my feet were even worse. So not only did the hike mangle my feet, but I forgot I was Irish for 20 minutes and took my socks off and was subjected to that this kind of sunburn. So when I say I've suffered for the science, I am telling the truth. But it has been worth it. It has been worth it because we have discovered so many really cool novel small molecule activators from these plants. And these are just some of the ones that we've found over the years. And there's a couple that I'm going to highlight today. Our operation has expanded somewhat. So this was taken from a figure that we published in a paper earlier this year, in which we did a 1,400 plus plant screen. And we started to hone in on what properties we want to find within these plants. So we have a KV 1.3 inhibition and KV 7.23 activation as our criterion. And this resulted in us finding some small molecules which helped with inflammation and also help regulate pain. So that's a brief introduction of what we do. Now let's get into the good stuff. So the first thing is a very quick overview of a discovery we made and published last year and how that ended up helping with addiction. So about 2023, we published a paper showing that rosemary extract modulates KC&Q channels. I should say that all the electrophysiology in this today is conducted with 2-electrode voltage clamp and xenopozoocyte expression system. And you can see here that rosemary opens casein Q2 and 3, robustly shifting the voltage dependence of activation to the left. And you can see big increases in current as well at hyperpolarized voltage. Very interestingly, and what really intrigued us was this really huge effect with Q3, Q5 heteromers. You can see really nice current increases here, especially at hyperpolarized voltages, but a really nice hyperpolarizing leftward shift in the voltage dependence of activation. So anyone who has read our papers knows that the next step was to ascertain what's going on here. So what we did is we screened all the compounds within rosemary, and we found this one here, carnosic acid. So carnosic acid is one of the primary constituents of rosemary. And what we found is when we screened it against our casein Q channels, we had some isoform specificity. You can see here, big activator of casein Q3, so nice increases in current density, especially at hyperpolarized voltages, and also that nice robust leftward shift in the voltage dependence of activation. Little activity at Q2, not too much, and nothing going on at casein Q4. A little bit of activation again with casein Q5, but this is the one I wanted to highlight, is casein Q35, the heteromer. You can see, again, really nice current potentiation at hyperpolarized voltages here in the pre-pulse, but then also a really nice significant leftward shift in the voltage dependence of activation as well, recapitulating that result that we saw with the rosemary plant extract. Now, this was kind of where the story stopped, because as I'm sure anyone who knows in the casein Q field, casein Q35 activators, well, where's Q3, Q5 expressed? Is it even expressed as a heteromer as EVO? Well, luckily, we have an assistant professor here at UCI, Kevin Byer, who studies addiction, and he found something really cool. He found that when he gave acute doses of cocaine to his mice, that casein Q35 genes were downregulated within neurons in the globus pallidus. So we said, oh, well, that's really handy. We've got a Q3, Q5 activator. Why don't you try that? So that's what he did. And what he found was that by giving high and low doses of carnosic acid to these mice, he saw a reduction in the cocaine-seeking behavior in the mice. So they were pressing levers, which gave them an infusion of drug. And when you gave them carnosic acid, this reduced it, you can see here, by quite a significant amount. And it also reduced the active lever presses. And this wasn't due to forgetting how to press the levers. This was due to the effect of the carnosic acid, which was really, really cool. And we're really excited about this. So this is just a quick overview of a compound which I'm particularly excited about, but wanted to tee up for the rest of the talk. And this was published in Neuron earlier this year. So the main bulk of this talk is going to be focused on ataxia. So I'm going to jump around with a couple of diseases here today, but this one is particularly exciting because we just literally got accepted last week, so we are looking to hopefully this will hit the shelves soon. So ataxia is a umbrella term for lots of different symptoms which occur. It's mainly associated with being a movement or disorder, but as you can see from this lovely bio-render picture here that I swiped, you can see that there's multiple different symptoms that are brought upon by this condition. It's relatively rare, or actually very rare. And it's also brought upon, in its name, it's episodic ataxia. So it's brought upon by stress. It can be diet, physiological stress, or even emotional stress that can bring upon these symptoms. We're particularly interested in episodic ataxia type 1. And this is caused by a mutation in the KCNA1 gene, which encodes KV1.1. Now, the majority of these variants are loss of function, but there are gain of function as well. But we're predominantly focused on the loss of function mutants. So we really got interested in this because of a collaborator, basically. So Rich Sidlow at the Valley Children's Hospital here in California, he's a genetic neurologist, and he specializes in, he gets referred a lot of cases which other neurologists struggle to diagnose and treat. And normally it's due to novel mutations in ion channels. So he comes to us to basically characterize these, and this has become a very fruitful collaboration between the two of us. And he came to us with this website. Sorry?
Here forever.
Oh. Someone seems. Okay, someone seems to be having get quite excited about this. So essentially, the KV1.1 mutant we got was an L155P mutation, which occurred in the N-terminus. And this was a mother and daughter mutation, so both had it, and they had they had clumsiness, staggered gait, and slurring of words. And they were responsive to the treatment with carbamazepine, but we wanted to see, well, is there any natural compounds that we've discovered over the years that could potentially help with this? Now, I'm jumping back in time a little bit because in 2020, we published a paper in JPET where we found that by modulating the structure of glycine by moving the fluorine around, we came up with a KV1.1 specific activator. And you can see here there's a really nice leftward shift in the channel's voltage dependence and also a big current increase as well. It had no effect at KV1.2 or casein Q23. So we were really excited by this because at the time this was one of the first examples of this specificity within the KV1 field. So we jumped and thought, yes, okay, let's try some KV1.1 linked ataxia mutants. We landed on trying this one here, E283K. This is from an Italian family, and this was characterized in 2017. And basically, the properties of this mutation are that it decreases current density and it also right shifts the voltage dependence by plus 15 millivolts. So we tried our glycine 4-TFMPG, which really rolls off the tongue there. And unfortunately, we didn't see anything. No increase in current density. We saw no rescue of the voltage dependent shift, and this is reflected in the there's no change in the resting membrane potential here. So we were a bit dejected by that. It made a nice story, but we really want to find things that actually help people. So Rich coming to us kind of reinvigorated our search. And this led to us going through the literature. And we came across some documented botanical ataxia therapies from the Kwakakewak First Nations people of the Pacific Northwest. If you want to read more about these people, you really should. Their history is really fascinating and also tragic. I won't go into it here, but I should say that their ingenuity is basically what was the springboard for what I'm about to show you. So they do deserve a lot of credit for this. But essentially what they found was that by using nettles, seaweed, and Pacific Nine bark, they could alleviate ataxic symptoms. So we decided, okay, let's see if these activate KV1.1. And we found, to our surprise, that they did. Then we wanted to know what was in there that was doing it. And we landed on gallic acid. So we found that gallic acid potently and efficaciously activated KV1.1. So we decided to try it against our ataxia mutants. So we L155P here, as I spoke about, and E283K. And to a happy surprise, we found that gallic acid really increases current density of L155P, which we showed was one of the properties of this mutant, one of its effects at reducing current density. And it also shifted the voltage dependence of activation. But also, it rescued E283K as well. It really robustly increased the currents at hyperpolarized voltages. And it also rescued the voltage-dependent shift that we saw. So it was 15 millivolts shifted to the right, as I said. Now with gallic acid at 1 micromolar, it's brought it back to that wild type. So this is fantastic. This is brilliant. And at the time, when we published this, it was the first example of this rescue of a KV1.1 linked variant. We found that gallic acid's mechanism of action is by binding to the top of the voltage sensor between the S1 and the S2. And we did a load of molecular dynamic simulations, which we backed up with mutagenesis because we found KV1.2 was insensitive. This was the mechanism of action of that compound. But as you can see here, we only rescued 2 out of the 5 mutations, which was great, but we thought we could do a lot better. So we put our nose to the grindstone, and we went back to the Native American people and their documents, and we found that these First Nations people also use pine tree extracts to treat movement disorders. which we would probably term more as diseases of hyperexcitability. So we went into the field and we obtained a load of different pines from a load of different locations. And what we found was that a lot of these pines activated KV 1.1. They either increased current density at hyperpolarized voltages or shifted the voltage dependence of activation in the hyperpolarizing direction. But we wanted to find something that did that, but without altering any of the properties. You can see here in a lot of the traces that even though these do shift the voltage dependence and increase current density, you see this big slowing of activation that occurs. And that may not necessarily be something we want for a potential compound for treating ataxia. And we landed on this guy here. So this is C. piscifera, or also known as false cypress. So you can see that there's big increases in the hyperpolarized potentials, but there's also a really nice robust leftward shift in the voltage dependence of activation. So this was the plant the pine that we decided to focus on. And just to show that I'm being everything I'm saying is true, you can see that we measured the change in activation kinetics. We saw nothing with C. piscifera compared to all the others. So our first port of call was, okay, can the plant extract do anything to KV 1.1 EA1 ataxia mutant? And we decided to look at V408A first, because this is the most well-characterized KV 1.1 ataxia mutation, and the only one at the time that had a mouse model. The characteristics of V408A is that it reduces current density, it shifts the voltage dependence to the right, and it also increases inactivation as well. So we put 1 in 50 C. piscifera or false cypress onto our V408A channel, and we saw, again, big increases in hyperpolarized currents, but also a really nice leftward shift in the voltage dependence of activation as well, where we saw mirroring that of wild type. So we've rescued two of these key attributes of this mutation with the plant. But what on earth in the plant is doing, right? There's no use just giving people the plant. I'm not going to reveal the compound name, because despite it being accepted, I'm still just going to play my cards close to my chest for now. So we'll call it metabolite Z. But you can see here, metabolite Z within false cypress robustly increases hyperpolarized currents and really dramatically shifts the voltage dependence in the hyperpolarizing direction in a dose-dependent manner. When you plot either V-half shift or the resting membrane potential shift as a function of concentration in metabolite Z, you end up with a EC50 of around 12 micromolar. And very nicely, you can see here, metabolite Z has no effect on the activation kinetics, akin to much like the plant from which it came. But very interestingly, it significantly slowed deactivation. So we kind of hypothesized that this might be stabilizing the open state of the channel somehow, but we still need to confirm that. So the wild type stuff's great, but what does it do with ataxia mutants? So what we did is we went to the genetic database and looked through the literature as well. And what we found was that there are multiple different EA1 linked KV 1.1 linked ataxia mutations. So what we decided to do was take several dotted throughout the channel to see if our metabolite Z is capable of rescuing mutations that occur in different positions. And what we found was really cool. So I have homomeric data for this as well, where it rescued about 6 out of 12. But in physiology, KV 1.1 is expressed in a heteromer with KV 1.2. So we decided to do the heterozygous EA1 mutant with heteromeric 1.1, 1.2 channels to mimic physiological conditions. And when we made these, co-injected these channels to form these complexes, we saw dramatic differences in the current density compared to wild type. We also saw significant voltage-dependent changes, often depolarizing shifts in the voltage dependence of activation, as shown here. And therefore, we decided to apply our metabolite Z at 12 micromolar. And what we found was really, really cool. Metabolite Z increased current fold density in all of the mutants, some cases as high as 15-fold, but then also had huge hyperpolarizing shifts in the voltage dependence of activation, so much so that for every mutation where there was a shift, it brought it back level with wild type. You can see here with E283K, the Italian family mutation brings that shift back beyond wild type. Really significant changes such as R307C here, which is found in the S4 domain, huge rightward shift. Metabolite Z brings that back in line with wild type, and the same again with V408A. So this rescued 12 out of 12, and this was fantastic. We were really excited about this, and this is a really cool finding. However, this is a heterologous expression system using TUVC and xenophazuicides. What about in vivo, right? This is the proof in the pudding. So we decided to generate a brand new mouse model. So we generated a E283K mouse using CRISPR, and we decided to put this through a battery of tests. The first test was the rotarod test. So the rotarod test, what we did is we put our mice on this rotarod, and it gradually started to speed up, and then we recorded, or it recorded, when the mice started to fall off. And you can see here that the wild type with vehicle and the E283K with vehicle are distinctly very different from one another. The E283K mice fall off much quicker than the wild types. And this makes sense, right? Because the episodic ataxia is brought upon by physiological stress, which this mimics. So what we decided to do was we gave each group 1 mg per kg of our metabolite Z prior to doing this test. And then what we found was that after giving 1 mg per kg 30 minutes prior to this test, both groups now look the same. You don't see any real difference between both E283K and wild type mouse groups, which was a really cool finding, and shows that metabolite Z rescues ataxia in vivo as well. We wanted to do a chemical challenge as well, and using the balance beam this time. And you can see here, when we just did our baseline levels, there's no difference between the wild type and the E283K mouse. So the hind missteps are when their horse slips from the balance beam. This makes sense because obviously neither mouse group, well, the E283K aren't stressed, so they should run along quite happily. However, when you give them an isopaterenol challenge, this is where we start to see differences. So wild type with vehicle, and isopaterenol, you see about 5 hind misfoot steps. However, our E283K mice with isopaterenol challenge and vehicle is almost double that. So our E283K mice, when stressed, are slipping a lot more. So when we gave the isopaterenol this time with 1 mg per kg of metabolite Z, what we found is the wild type relatively no change between that and vehicle, which is good. But this time, E283K mice with 1 mg per kg of metabolite Z are back down to comparable to wild type levels. So now their slips are the same as the wild type mice. So we believe this is unequivocal evidence that our metabolite Z is rescuing ataxia in this mouse model. I've included p-values here. I don't like p-values personally, but they're there for the journal. So just to show you, hopefully this works, we have some videos. So the first video I'm going to show is vehicle plus isopaterenol with our KV1.1 E283K mouse. So you can see here, the mouse is struggling to get across the balance beam. It's slipping quite a lot. We have to help them up sometimes. You can see that they're very slow at doing it as well. This is the clumsiness involved in it. So you can see not completing the task very well at all. When you compare that to the wild type with vehicle and isopaterenol, you can see that the mouse is much more actually crossing the balance beam, very little slips, much more confident at doing as well. They get a little bit bored sometimes, so this guy is going to shoot round and try and turn back on himself, but we have to politely encourage him to keep going. And you can see that he gets across just fine, albeit taking his time. Right. Now, what about when we add metabolite Z into the mix? So metabolite Z plus isopaterenol now with our 1.1 E283K mouse. And you can see here, remember before it did not cross very well, it slipped a lot. Now we've got very rarely slipping, much faster at crossing, less clumsy. And then just to show you that the same combination in the wild type mouse has no effect either. So unfortunately, this one's going to the bathroom. I should have picked a better video. But you can see this mouse, again, it's wild type, so very few slips. But the main point is between the two, the mutant and the wild type, there is now no difference now that we have added metabolite Z. Yeah, so there you go, and we are really excited about this, and I think it's a really cool finding. But how does it work? How is this happening? Well, this is where we bring in some very clever people. Assistant professor here, Alvin Yu, specializes in all atom molecular dynamics simulations. And what he did was he constructed a KV 1.1 channel using AlphaFold, and did some molecular dynamics simulations to see where gallic metabolite Z binds. So you can see here we placed it in the extracellular solution. So we're not putting it in the channel. So this is where it just finds its way naturally. So you can see here, metabolite Z goes into the cleft of the voltage sensing domain of the channel. He modeled the whole channel, but I'm just focusing on the VSD here just to give clarity. And you can see that metabolite Z sits in that cleft very happily, sticks there very nicely, and it doesn't want to move. These simulations are about 100 nanoseconds each, so they're quite long. You can see here, beautifully situated. And then here's metabolite Z with the complete tetramer in the lipid bilayer domain. Again, you can see it's sitting in the top of that voltage sensing domain. And this is just a static of what I just showed. So what we're seeing is that metabolite Z is entering a cleft in the voltage sensing domain, which is causing, we saw a compaction of the voltage sensing domain, and it also caused a tension on the S4-5 linker, which basically it was co-opting, metabolite Z was co-opting the voltage sensing domain to basically mimic what would happen with membrane depolarization in order to open the channel. And what we found is when we looked at the retention time of metabolite Z in our model, we found that it was situated at a R295, K193, and E187. And when we mutate these out of KV 1.1 to alanines, we see a really significant reduction in the effect of metabolite Z on KV 1.1. So that is how this works. So we've gone from the all atom aspect of how metabolite Z works all the way up to the mouse model to show that it rescues ataxia. And this should be coming out very soon. This is really exciting stuff. And I'm always mesmerized by what these molecular dynamics guys can do. It's far above my head, but it looks fantastic and it tells us a lot. And now, in the words of Monty Python, something completely different. I'm going to talk about top channels in fungus to end, and how we discovered that they are regulated by extracellular pH. So fungus are very important. We need them for helping prepare foodstuffs, very important for alcohol, which I'm sure we can all agree this time of year is especially important. We have found many antibiotics and so forth from fungus, penicillin. And about 450 million years ago, fungus were really important for the migration of plant life from the aquatic to the terrestrial environment. So suffice to say, without fungus, we really wouldn't be around here today. That's to say that not all of them are very helpful. There are pathogenic fungus, and sorry again for these pictures, but some are human pathogens, so they can range from the mildly aesthetically unpleasing athlete's foot and oral candidiasis to much more serious bloodstream infections, which can be fatal, especially among the immunocompromised population. In fact, fungal infections in humans kill more people than tuberculosis and malaria combined. So they're a really big problem right now. They also affect plants as well, so phytopathogens. So they decimate lots of crops which are important for the calorific needs of the global population, such as rice blast and wheat blot. And in really significant cases, it can cause famines, such as the famine from my country, the Great Famine in the 1800s, which still has ramifications today in Ireland. Um, they can also be quite weird and wonderful. There's the cordyceps fungus as well, or the zombie fungus, which basically infects its host, these insects here, and it's this part of its sporulation process, and it controls the unfortunate insect. This was used for dramatic effect in the HBO series The Last of Us, where it was shown to do a cross-species barrier and infect humans and bring about the downfall of civilization, creating these horrible zombie-like things. If you haven't seen it, it's really good. But I'll stop plugging HBO for now. So why are they big problems? Well, mainly because fungus are very, very clever. They have developed a lot of resistance mechanisms over the years, and they do it very rapidly. Another issue which is associated with this is that the number of antifungals we have at our disposal are very few and far between. And the ones that we do have are already showing resistance and often do so very quickly. And then the other problem is the shared homology between host and pathogen. So a lot of these antifungals do their business by punching holes in the cell wall of these microorganisms. And unfortunately, they're not very good at discerning between the fungal cell membrane and cell membranes of of human tissue. So you get lots of nasty side effects, which in cases like amphotericin B can be fatal. So we need new molecular targets to develop antifungals. And this is where TOC channels come in. So TOC stands for two-pore outwardly rectifying potassium channel. It's comprised of 8 transmembrane domains and 2 pore loops. Closest cousin to it, and I use the term cousin in inverted commas here, because is K2P channel, which I'm sure we're much more familiar with. But I'm sure you can see that the topology of these two proteins is very different. Biophysically, they're very different as well. So TOC is strictly outwardly rectifying, whereas K2P channels, you do get linear conductance. These were first discovered 30 years ago by the Goldstein group in Saccharomyces baker's yeast. And they showed as part of that work that if you modulate these channels, it can cause unregulated potassium efflux, which results in or triggers apoptosis and cell death. There's no homologs in humans, animals, plants, or insects. So specificity is kind of key. And then, but that was kind of where it stopped until Tony Lewis and the team that I was fortunate to be part of in a very small way. But Tony Lewis cloned TOC channels from lots of different human fungal pathogens, and including this one here, Candida albicans, which is a really big issue in hospitals right now. It's one of the main hospital-acquired fungal infections. So they look like a promising antifungal target, right? There's specificity. If you modulate them, they can kill the fungal microbe. But we don't know very much about how they're modulated and what they do in fungal physiology. So what we all looked at was, well, we all have Candida within us. And normally it's very happy in its commensal yeast form. But there's lots of different environmental triggers that can turn it from this commensal yeast into a hyphal form. And we decided to start testing some of these to see whether these environmental cues could modulate TOC channels. And we landed on pH. And what we found was that Candida albicans TOC, or CUTOC, when exposed to extracellular acidification, dramatically increased current density and also shifted and caused a hyperpolarizing shift in the threshold of activation. Very interestingly, alkaline conditions didn't have much of effect. I should say that the switch from acidic to alkaline conditions is what triggers hyphal growth in Candida albicans. And you can see here that the onset of action is very rapid. So you switch from neutral pH to pH 5. This opens up the channel. And then when you go back to neutral again, it goes back to baseline. So when you plot things such as current density or threshold of activation shift as a function of pH, you get a pKa of around about 6. We also tested other TOC channels from Aspergillus and Cryptococcus and Saccharomyces. And what we found was that none of these were pH sensitive. So this pH sensitivity appears to be very specific to Candida albicans TOC. So armed with this information plus the pKa of 6, which is akin to a histidine residue, we decided to do a sequence alignment, and we found that there was a histidine conserved in the S1, S2 linker of the channel, which was not found in the other top channels. So we decided to mutate this to see whether we could get rid of the pH effect. So first we mutated it as a lysine to mimic acidification. And you can see here that while there's still a current increase, it's not as pronounced as before, and that the hyperpolarizing shift in the threshold of activation is also mitigated as well. When you mutate that histidine to an asparagine, so a neutral residue, we see this same reduction in affecting current density and hyperpolarizing shift as well. And you can see when you plot that as a function of pH, it looks dramatically different from wild type. But it's not the full story because we haven't got rid of it completely. So we decided to look for the other part of this pH sensor in this ion channel. One of the phenomena we noticed was that when we exposed the Candida albicans talc to acidic pH, it shifted the reversal potential of Catalc closer to that of EK. So, and which also the mutation of H144 to a lysine did as well, mimicking acidification. This also shifted the reversal potential closer to EK. So it suggested the potassium channel, or the Catalc channel, I should say, was becoming more selective for potassium, and this was confirmed the pseudo-bionic substitution experiments. So it suggests that the pore domain is involved somehow. So I'm not going to go through all of this due to time, but essentially what Tony and Steve Goldstein's group did is they constructed these chimeras using Catalc and the pH insensitive SC talc, so Saccharomyces talc. And what we all found was that when you mutate this, when you swap the second pore domain of Catalc with SC talc, you significantly reduce the current density increase. And what we localized it to was a serine here at position 466 in the S8 domain, which when you mutate that to an isoleucine, which is the corresponding amino acid in SC talc, it gets rid of this current increase caused by extracellular acidification. Very interestingly, it doesn't have an effect on threshold of activation. You can see that S466I is still almost as same as wild type. You see here the histidine, when we mutate that to an asparagine, significantly reduces this effect. But we found another residue here, so V462, within the S8 transmembrane domain. Also, it didn't have much of an effect on current density increase, but it did significantly inhibit the threshold of activation. So what we decided to do was we created a triple mutant in which the histidine, the valine, and the serine were all mutated to the neutral residue, and then the two corresponding residues in SC talc. And what we found now was that the Catalc was no longer responsive to extracellular acidification, but also this threshold of activation shift that's caused by acidic pH is also reduced to a negligible amount. So it appears that the Catalc voltage sensor, or not the voltage sensor, the pH sensor, is formed of a tripartite of amino acids. You have the histidine in the S1,2 linker, so it's sat here, and then you have these two residues within the S8 that are also form this pH sensor, and it appears there's some sort of crosstalk or coupling between the two, which has been shown previously by Dan Miner's group, I believe, in mammalian K2P channels. But how this works, we're still trying to work this out. So I believe that's everything from me. Ooh, no, I'm not going to show this stuff. This is something else. So I just wanted to thank everyone. Professor Jeffrey Abbott, who has been my long-term mentor and a great friend. Ryan Yoshimura, who did all the behavioral studies that we showed here today for the ataxia. Dirk Hogenkamp, who's our medicinal chemist. He makes lots of different analogs of the compounds we find to try and improve potency and efficacy. Our army of undergraduates who help process the truckloads of plants that we collect on these adventures. I wanted to highlight three in particular. Claire Illich, who was fantastic in helping with a lot of these experiments, particularly the pH talc channel story. And she's published quite a few papers now. She's now a grad student here at UCI. On Nguyen, who has got some papers coming out soon, who's just a phenomenal undergraduate, and she's just been given a flash talk at biophysics. So please do attend that. She's really one for the future. And Amy Nguyen, who diligently scored the hind foot slips of the mice in the ataxia study. So these are going to be three names that I'm sure will come up on be giving their own EpIC seminars in the future. The Alvin Yu lab, who did all the molecular dynamics simulations, and Lorenzo is grad student who contributed as well. Kevin Byers' lab here at UCI for doing the addiction study stuff. The Goldstein lab and particularly the Lewis lab in Portsmouth for the pH tox studies. They were instrumental in this as well. And Tony Lewis was my PhD supervisor, so he was instrumental in where I've got to today. Rich Sidlow at the Valley Children's Hospital, a great collaborator who we've got some exciting stuff in the future coming out with. The various national parks, the Kwak First Nations people. Without them, I'm certain we would not got where we did with the ataxia story. So thank you to them. And also all the funding bodies as well who've made this work possible. And in particular, I'd like to highlight the Susan Samweli Institute here at UCI, who really backs us with this alternative, this medicinal plant stuff. And you know, without their support, we surely we wouldn't have got as far as we have. So and thank you all for listening to me for the last 45 minutes.
Brilliant, Rían. Thank you very much. You know, I'm a fan of your work. I think this is really inspired stuff, and that was a very entertaining talk, despite the feet pictures. So thank you very much for the for the guidance. We have a few questions in the Q&A, so maybe I can start there. We've got a bit of time here. So Elizabeth Serbai asks, any effect of metabolite Z on heart rate?
We haven't looked at that. So this has basically just been focused on ataxia, but our next phase of these investigations is going to start looking into how this affects the wider physiology, because we have a lot of hope that this could be used as a druggable compound. So we are starting to collaborate with some other people in order to look at these different physiological aspects. But I to say right now, I don't know. I hope not.
Mark Rogers says, lovely talk, Rían. Did I miss you saying anything about the KV1.X subtype selectivity of the Pacific Coast ataxia botanical extract, so subtype selectivity? The S1 sequence comparison seems to show high amino acid conservation across the isoforms, particularly interested in KV1.3 due to its expression in T cells, and KV1.5, the atrial isoform, as drug discovery targets.
Yeah, so sorry. In doing a rapid overview of all the different things we were doing, I didn't get to focus on that as much as I'd like. Those particular extracts don't activate KV1.2, and they don't. Well, actually, maybe Nettles does, but gallic acid within them. The important thing is gallic acid within them doesn't. And that discrepancy between gallic acid for 1.1 and 1.2 was what it led us to find the binding site. What these extracts do at other KV1 channels, I don't know. That would be really, really interesting. I showed in that first few hundred slides ago that we use KV1.3 inhibition as a screening parameter. So we're very interested in extracts that do that for obvious reasons regarding inflammation and so forth. So what these do at other KV1s, I don't know, but that would be a really interesting avenue to pursue.
Okay. Julius Ashok-Kozak asks, could you repeat, please, what was the explanation for the reversal potential change due to pH change in the TOC channels? Do TOC channels express in transfected mammalian cells too?
So which slide were we on? The change in reversal potential. So basically, the change in reversal potential was essentially when you switch to acidic pH, what we saw was that it was we saw the shift become more hyperpolarized, going closer to EK. And what we found was when we hypothesized that this was because the TOC channel, when Tony Lewis and co first characterized this, they showed it wasn't perfectly selective for potassium. It also passed sodium and other ions as well. So the pH effect essentially is changing the selectivity filter to make it more selective for potassium. So the reversal potential is now becoming more hyperpolarized.
And it's not just simply that there's very little channel activity around the resting membrane potential prior to activation of the channel.
So yeah, so top channel activation is coupled to EK. Once you pass the threshold of EK, then top channels become more active. It looks that, I don't know whether I mentioned, these don't have a voltage sensor. It looks like they're voltage sensitive, but because of it, that would not be good for them in a fungus. So once you pass EK, these top channels become more active. So it is around EK that they start to open.
Thanks. Damien Bell says, always fantastic work. Congratulations to you, Jeff, and all your collaborators. Your compaction of S4 in KV 1.1 by metabolite Z strikes me as another potential mechanism for voltage sensing in KV and NAVs and CAV channels. Do you think this is almost mimicking what happens in voltage dependence, or could this be a true alternative, i.e. the translation we normally use as a model is actually a compaction effect of voltage?
That's a really good question. So when I said that it was, let's see if I can find the slide here. Yeah, so we said it was co-opting the voltage sensor, and as Damien pointed out, this compaction effect, it showed that it made it more energetically favorable which would suggest that it wouldn't need as much of a push to open the channel. So this could be a new mechanism, certainly could be. We've had to be very careful with the language. We do use ligand binding and activation in the paper. I'm not sure whether that's going to cause any contention, but yeah, so we do believe this is a potentially new mechanism. It certainly could be. It needs a lot more investigation as well. So it is very exciting, and just the power of what people like Alvin can do with these molecular dynamic simulations are just really opening up being able to investigate these things a much further.
Damien also asks the more important question, something he's asked Jeff previously: do you need someone to carry bags on your trips? He'll bring the sunblock and a spare tent.
Oh, if you could have carried my backpack, that would have been grand. I wouldn't have nearly passed out on the beach. Yeah, I think Jeff said, as long as you've got a good rum tolerance and beer tolerance, we're always up for additional members of our expeditions.
A question from an anonymous attendee: could metabolite Z have an effect on other channels, sodium and calcium channels, for example?
I mean, it could. We haven't tested it. We've tested it against KV 2.1 and HERG, Q2, Q3, and it did nothing. But there's always the possibility that, as we all know, we like to say specificity, but it's specificity within the confines of what we have tested. There is always that potential that can modulate something else. But yeah, in answer to your question, I don't know, but it very well could do.
We've got a question from Bryce DeRocha, sorry. With the specificity of pH sensing to C. albicans TOC, have you looked at the variation between different TOC isoforms of C. albicans due to non-synonymous variation between different isoforms, I suppose? Yeah,
so we haven't been able to clone any other TOC channels from any other Candida species yet. It's something we're looking into. We just worked with what was there historically through the amazing work that Anthony Lewis did, getting these original ones. It would certainly be very interesting. I know Candida auris is a big deal right now. So if that does have a TOC channel. So that is something to look at in the future. With TOC channels, we've only really been looking at these as within the last 18 months. It's not really been a focus of the lab at all. So this was a kind of a side project, but it'd be certainly something interesting to dive into in the future. But that discrepancy between pH sensitivity is really interesting as well, because we believe it's something to do with the environments that these Candida microbes inhabit compared to the other ones, where changes in pH are much more dynamic.
Excellent. I think we're getting to the end of the Q&A questions. Any more? Feel free to add them at this point. I've got a general question about the whole philosophy, Rían. So you take these plant extracts. So we've seen many successes of this approach, How what maybe is missing, and we discussed this briefly beforehand, when you identify the plant extract and you see an effect of the crude extract, how much work is it to always identify the key component? Is it frequently the most prominent component in the extract? How do you go about narrowing down?
Yeah, that's a really good question. It's not always the most abundant component. I really wish it was, because then my screens would be a lot quicker. We've previously been doing it just by sheer brute force. So we would look through the literature, see what's been already characterized within these extracts, or in cases where that hadn't been the case, we do mass spec here at UCI with our chemist Derek Hogenkamp. Yeah, it's not always the most abundant compound. So it does take sometimes screening of up to 20 different chemicals before we start to find a hit. We've got hits, really exciting hits of different extracts where we still haven't been able to ascertain what the active metabolite is, and we've screened in excess of 20 different components. So yeah, I know I think we were saying it looks like we're always just like, you know, ding 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 all the time, but there's a lot still sitting on the bench that we just haven't worked out yet.
And do you ever test it in combination then if you don't get a hit from the isolated?
Yeah, so we had a paper 2018 where we had two active metabolites within forget the name of the plant now. It was there was malatoxin and isovaleric acid. Essentially, we found malatoxin as an active metabolite, but it didn't explain the anticonvulsant effects observed with the plant that a previous group had shown. So then we found isovaleric acid in there, also activated Q2Q3. We found that when we combined them, they had this huge synergistic effect. And then lo and behold, when you combine them together in the anticonvulsant study, it then became an anticonvulsant. So that's the only instance we found so far. I'm sure there's others out there.
Brilliant, thanks. John, would you like to ask your question?
Oh, yeah, no, I mean, also just your whole way of doing work and finding these compounds is just incredibly cool. And I just big love. And I was curious about the metabolite Z. I was noticing that when you were showing kind of a dose response, that as you kept applying more and more of it, if I saw that right, it keeps having more and more powerful opening effects. Is that?
Yeah.
And I was just wondering how, do you have a way that you're thinking of that? How that could be? It looks like from this point of view, an unsaturable agonist, potentially a full agonist at some concentration.
Yeah, so one thing I didn't show, but we do show in the paper, is that when you start to get to concentrations beyond 30 micromolar, you do start to see a suppression of the pre-pulse current, which is where we're starting to see. So this happened with niflumic acid as well, which was another paper that was published in PNAS last year. The concentrations used there were in hundreds of micromolar. So in order to get their effect. So we're already dramatically improved on that. But yeah, so we think that this effect occurring here at 100 is also partly to do with you're seeing suppression of the pre-pulse current as well, which is obviously not something you really want. So we're also at the mercy of what we can solubilize it in. So DMSO. So we'd like to go. It would be nice to find that saturated dose, but obviously you've got to keep in mind, the percentage DMSO in the bath going. Yeah. So it is really interesting. No, I agree. It is really interesting. It's one of these metabolites that is just kind of came completely from left field and has been great to work with. So we hope to answer more of these questions. Hopefully, we can make a more soluble version as well. That would be fantastic, or something that's solubilized in something a bit more malleable than DMSO.
Yeah. Do they wash out, or do they just kind of?
Yeah, they do. They do wash out. Yeah, they wash out very quickly, actually. So interestingly, metabolite Z washes out very quickly compared to one of its analogs. I did wash in experiments. Because as pharmacology, that's your gold standard, and it's nice to look at as well. But yeah, they do wash out relatively. Metabolite Z does wash out relatively quickly.
Cool.
Which kind of fits the binding site, right? Because it sits at the top, so it should be more easy to knock off.
What's the concentration in the MD experiment? And then we'll go to Layla's question.
Oh, that's a very good question. Yeah. You know what? I'd have to ask Alvin. Yeah, I've forgotten that. Yeah, no, that's a very good question.
Rían, that's really, really beautiful work. I was really impressed with your mouse data, and I was wondering, kind of related to the last bit of this discussion here, like how do you administer metabolite Z in the in vivo experiment, and how long does the effect last?
Yeah, so it's an IP injection, and it's administered 30 minutes before we run the experiment alongside isopaternal. So how long it lasts, I don't know. That again is going to be the next phase. So when we're working out what does it hit, what other physiological effects does it have, that is half-life is going to be another big question to ask, because obviously you're right. If you have ataxia, you don't want something that you have to keep repeatedly taking every couple of hours. You want something which is a slow release and doesn't get broken down so quickly. So yeah, that's something we're going to look into and in earnest, certainly.
Thank you.
Rían, we're over the hour, but if you'll indulge us with one more question from Harold Zakon on the Q&A, because it's something I was wondering as well. So what he asks, what are the benefits to the various plants that have these chemicals? And maybe you can comment on what these plant-derived molecules are doing in vivo in the plants. Are they all plant sterols or alkaloids?
Yeah, some are flavonoids, some are alkaloids. This is a question that we're intrigued by as well. What on earth are these things doing? I don't know enough about plant physiology, unfortunately, to kind of hazard a guess. I mean, all these plants have various ion channels and things as well, so it's not beyond the realms of possibility that these molecules signaling molecules as well. So yeah, maybe they do have beneficial effects, or maybe someone has already discovered, maybe what gallic acid does in seaweed or something like that. But yeah, to my knowledge, I'm not entirely sure. But it's a really interesting question, right? How this stuff evolved to be, and we've now utilized it. We had a discovery a few years ago in PNES where we found that antihypertensive or herbs used as antihypertensives, their unifying molecular mechanism was via casein Q5 activation. Why on earth that came about, I don't know. But it's evolution at its finest, I guess.
Fantastic. Okay, on that note, I think we'll wrap up. Thank you again for the presentation. That was fantastic. And thank you everyone for attending. We'll see you next month.
Cheers.
Thank you.